Everything is good to go here. I have this set up. Perfect. Okay, so let's do a quick introduction. I'm sure you guys have all gotten my emails, but today our presenter is Dr. Tara Palmore. She's an infectious disease specialist and deputy hospital epidemiologist at the National Institutes of Health Clinical Center. In her clinical role, she serves as an infectious disease consultant in the clinical center, which has a large stem cell transplantation program and other categories of heavily immunosuppressed patients. Dr. Palmore did her undergrad education at Harvard College and attended the University of Virginia School of Medicine. She completed internal medicine residency at the New York Presbyterian Hospital, Cornell, and Infectious Disease Fellowship at the National Institute of Allergy and Infectious Diseases. So let's go ahead and get started. Dr. Palmore, the floor is all yours.
Great, thank you. Thank you so much for inviting me. I appreciate the opportunity to present to you, and I'll be talking about transmission of antibiotic-resistant bacteria from hospital drains. But I'll go a little further than drains, as you will see. Let me just make sure that I can control the slides. Am I controlling these?
No, I am controlling these.
Oh, you're controlling them. Okay, all right. I'll just tell you when to go to the next slide. So my major disclosure is that I'm going to get really down into the weeds of the plumbing here, which is why I have this picture, to remind you that we're really going to be talking about plumbing, including toilets. So let's just get into it. Next slide, please. So these are the objectives of the talk. First, to understand how drain and wastewater-related organisms can threaten patient safety in the healthcare environment. And second, to learn about strategies and tools to identify and mitigate drain and wastewater-related infection risks in your facility and healthcare facilities in general. Next slide, please. So I'm going to start with a real case, and this is a case from our hospital. So first, just a brief intro about the NIH Clinical Center, which is in suburban Maryland, right outside Washington, D.C. So we are a 200-bed clinical research hospital at the NIH, and everyone who comes to our hospital is on a clinical research study, and most of the patients are really very sick, often getting experimental therapy for conditions that they've either run out of options for or there's no treatment for in normal hospitals. So this is a case of a 23-year-old man who was in our hospital because of a congenital immunodeficiency, and was at the time of this case was hospitalized for complications of a stem cell transplant and had a newly positive perirectal surveillance culture. So in our hospital, we do surveillance. We do perirectal surveillance for carbapenemase-producing organisms. So this man's surveillance culture grew a KPC-carrying organism. It was a KPC-positive Leclercia attacarboxylata. Well, you all know what that is, of course. No, I'm kidding. And I didn't know what that is, and I was very happy because our microbiology lab director didn't know what that is, had never heard of this organism, so it made me feel better. And it turns out it's an unusual species of Enterobacteriaceae. So it is an enteric organism. It's just really unusual. And the fact that it's so unusual was actually helpful because it made us really take note. And it was KPC carrying organism. This patient had actually had only negative surveillance cultures in the last 18 months that he'd been receiving care in our hospital. At the time of this case, he had negative blood and urine cultures, so this was only colonization at this point. Next slide. Now, I'm telling you as if it were a surprise, but there is actually a backstory. This wasn't the first time we had heard about this organism. And in fact, the backstory starts 3 months earlier when one of my infection preventionists cultured the drain, this drain right here to the right, in one of the housekeeping closets. Now, all healthcare facilities have these closets, and they're the locked rooms at the end of patient care wards whether they're in long-term care facilities or in hospitals, acute care hospitals, or community hospitals. And this is where mop bucket solution is dumped out into a floor drain, wax, after floor waxing, and that's what a lot of this residue that you see in the drain is probably wax and all of the nastiness that gets stuck in wax. And why did my infection preventionist culture the floor drain? It was actually part of a research study that we were doing to study the microbiome of the wastewater sites in the hospital. And this is a good story about be careful what you look for, because then you have to deal with what you find. So this is what we found. 3 months before this patient's case, we cultured this drain and we found KPC Leclercia carboxylata. And that was strange. Look, we weren't surprised to find a KPC organism in drains, because as I'm going to tell you, drains in hospitals, whether it's our hospital, your hospitals, are full of resistant organisms. But this was an odd one. So we took note of this, and then next, please. So the next thing that happened was, so don't click again. The next thing that happened was cultured 4 more environmental service closet drains. And they're nasty. I mean, the biofilm is not subtle. You can see that here. These don't get cleaned in any way. So 4 different closet drains then a month before the patient's case now grew this same organism. And now we actually really started getting worried. There's no handbook of what to do when you find the same unusual organism in environmental service closet drains, because it's just not normal to do these cultures. But we knew that there was something bad happening here. And just as we were working to figure out how to go about investigating this and what this really meant and whether it was a patient safety risk, then next slide, please. Then the patient turned up with the organism. So obviously we did a big investigation. How did the organism get from the floor drains to the patient? And because we had been doing this research study and then, following it up, we knew for a fact the sequence of events. We knew that it had been in at least one closet drain, and then multiple closet drains, and then got to the patient somehow. So our investigation involved culturing dozens of environmental sites, including surfaces in the patient's ward, other housekeeping closets, and surfaces other than the drains and housekeeping closets, and then environmental services equipment, including these mop buckets, which look like this. And we actually found in one of the 7 mop buckets that we cultured, we found the same organism. On the right here is a phylogenetic tree of the core genome sequencing that was done in a genomics lab, on microbial genomics lab at NIH, in which they compared our sequences to those of the some outside Leclercia species and isolates. And so let me try to walk you through this. I don't think you can see my mouse since I'm not controlling the slides. So in the top box, so basically the genomes clustered into 3 clusters. So in the top red box that's labeled A, patient, where it says patient V, PTV, that's where those are the patient's isolates. And they're 2 different, probably swabs from the same patient. And where it says housekeeping equipment, that's the bucket. And so what that means is that basically the patient and housekeeping bucket isolates were a near match. They weren't an exact match. They were a few SNPs or a few nucleotide variations apart. So they weren't totally identical, but they were extremely close, suggesting that that was either a direct transmission or a very close indirect transmission from the bucket to the patient. And how could that have happened? Well, you all do this for a living. You can easily imagine floor cleaning solution. The floor gets covered with cleaning solution. The patient drops a pillow or a banana and picks it up and ingests a few bacteria. And a patient like this, who's immune compromised and has been on antibiotics, has a completely deranged gut microbiome and easily becomes colonized with resistant organism, even just a few bacteria. So that's cluster A. Cluster B was a few of the closet drains, which are genomically slightly distant from the patient and the bucket, which suggests that there wasn't immediately direct transmission from the drains to the bucket, and that makes sense given the amount of time that passed from when we cultured the drains to when we cultured the bucket and the patient. And then the third cluster at the bottom, which is C, was other closet drains, which are slightly different. You can see they're on a different level than this isolate labeled biopsy, which was an outside specimen that we got from another hospital. And then there's also some outside, the ones called livestock and drinking water and lake water. And that was just to show that other Leclercia isolates that were unrelated to the ones in this little outbreak were, looked different genomically. So this was an event, and this was really taught us that that what's in a drain can reach a patient if there is bad adherence with housekeeping practices. And what we discovered in the course of this, you can imagine we discovered a lot of housekeeping practices that were unfortunate, that led to organisms from the drain getting into a bucket, then to a patient. And so all kinds of things like, the hose that fills the bucket getting placed down into a dirty bucket to fill it up and rinse it, and splashback from the drain likely reaching the bucket when the waters run, and all kinds of things that were accidental and some that were due to poor practices. But one of the major things that had happened was that, unbeknownst to the infection control department, the environmental services had switched out our disinfectant floor cleaner for a neutral floor cleaner without ever telling us. And that had happened a couple of years earlier, and we had no idea. So that was news to us, and probably this was all preventable if they hadn't done that. So lots of interesting lessons learned there. So next slide, please. This is more about our research study, looking at the microbiome of what we can call the bowels of the hospital, the drains and the wastewater. So we sampled 128 sites in environmental services closets, and 12.5% of those samples grew carbapenemase-producing organisms. And you can see that apart from the Leclercia, there were a lot of other things. And most of these species that you see listed here, we had never seen in patients, even though we do surveillance for carbapenemase-producing organisms. On the next slide, we also sampled sink drains. We sampled faucets. We didn't find them in faucets. That's very reassuring. We found one carbapenemase-producing organism on an aerator, when that's probably, again, because of splashback from a drain. And then all of the other 10 of the 11 isolates that grew carbapenemase-producing organism were from the sink traps and drains. On the next slide, we even went into hospital sewage pipes and the manhole on the campus, right outside the hospital, and what I can only describe as sludge. As you might imagine, most of those samples grew carbapenemase-producing organisms. And again, many of these species we had never seen in patients, and it likely represents the KPC plasmids, mixing it up in the very rich biofilm of the wastewater pipes with species, and they're very promiscuous, and there's a lot of they do a lot of partying down there in the pipes. Next slide, please. So there is a wonderful review that came out at the end of 2018 by Philip Carling in Boston, which is a review of carbapenem-resistant organism outbreaks that are related to wastewater drains. And most of these actually relate to sink drain-related outbreaks. So people have been reporting in the literature for about, 25, 30 years, hospital outbreaks, mostly ICU outbreaks related to sink drains. But until the last probably 5 to 10 years, you really couldn't tell whether the isolates in the sinks and in the patients, which way they went, whether there were just a lot of infections in patients, say with Pseudomonas, that were transmitted from person to person, and then the Pseudomonas went into the sink drain, and so then the investigators found it in the sink drain, or whether it actually went from the sink drain to some patients. You couldn't put the direction on the arrow. But now in the era of microbial genome sequencing being readily available, you actually can put the direction on the arrow, just like we did with our housekeeping closet drain. So because of that, there are now really good outbreak reports, and Dr. Carling assembled them. What he found in doing this essentially meta-analysis is that that in these 23 outbreak reports, there was a whole range of interventions, and they involved a total of 344 patients. So some of these, were pretty small, and some of them were bigger. But every type of intervention was described here: pouring disinfectants down the drains, scrubbing sinks and plumbing, replacing sinks, replacing plumbing. What we did with our housekeeping closet drains, actually, I didn't even tell you, we went through the hospital over a period of a couple of months, and with environmental services and facilities people wearing Tyvek suits, just to avoid kind of dirtying and contaminating themselves with resistant organisms. And they scrubbed out and used bleach and wire brushes to really scrub out and try to clean as much and as deeply those drains as possible. And then bleach and use hydrogen peroxide vapor to disinfect as many of those closets as possible. It was an expensive and resource-intensive solution, but we never saw another case of, or even another isolate of that Leclercia after we did that. But that type of intervention was described in here, not ours actually, but that type of intervention was described in this report. The problem is that in this meta-analysis, outcomes were not well documented. And in fact, part of that may be because nothing actually really works that well. So in our closet drains, next slide, in our closet drains, they did become repopulated fairly quickly with other resistant organisms, just not the Leclercia. There is nothing that you can pour down these drains, nothing you can do, stick down the drains, nothing you can clean them with on a temporary or ongoing basis right now that will really keep organisms at bay. Spraying bleach down them, pouring hydrogen peroxide down them, cleaning with wire brushes, the organisms come back because they come from below and they come from above. So this was actually an outbreak report from the United Kingdom that was reporting a very long and very large outbreak of carbapenem-resistant E. coli, most of which was KPC E. coli, in a cardiac hospital I think it was in Manchester in the United Kingdom. And most of it involved sink drains, but some with shower drains. that were documented to be colonized with these organisms on the most heavily affected wards, on the wards where the most patients were affected. And just to give you an idea of the scale of this outbreak, it lasted for over 6 years, at least at the point at which they published it in 2018, and involved 514 patients who were either infected or colonized, which really just, 514 patients, and I think there were 340 patients described in 23 studies in Dr. Carling's paper. So this is all in one outbreak. It's just enormous. On the next slide, you can see that so this looks complicated, but let me break it down. On the top, this is really a timeline of their patient epi curve and their interventions, and it's quite remarkable. So on the top here, you can see the epi curve and see where their black arrow is. Yes, thank you. This is their patient epi curve, and this is in A, which is the top part of the graph. And you can see that it all started really in January 2014, and those are the number of patients with their first positive carbapenem-resistant cultures. On the bottom, on B, are the interventions that they tried. And you can see the list of interventions. And they tried everything that you and I would have tried. Environmental screening, weekly and then twice weekly patient screening, staff cohorting, patient cohorting, closing. They even closed wards multiple times. Look how many times they closed different wards. Like one ward they closed 5 times, one ward they closed 7 times. And none of that, as you can see from the epi curve, which is on the same timeline as the interventions below, none of that worked. The only thing that appears to have worked is, see where there's where it says plumbing replacement. And actually, if you click, I think I have some little boxes and stuff. If you click ahead one. Yeah, thank you. And one more. Plumbing replacement is the only thing that appears to have had any effect. And so when they ripped out a bunch of plumbing, you can see that there was a pause in the epi curve. And when they did that, the rate of new infections or in colonization slowed greatly. But it didn't stop. They still had cases. And actually, at the time of publication, it looked like they were on their way up again. But which just tells you how very tenacious drain colonization and patient infections and colonization from wastewater sources really is. So I'm just telling you everything bad, like as if there's no solutions here. But we'll get to solutions. Next slide, please. Okay, yeah, I said this. So I'm showing you now. You can click ahead. Oh, so yeah, one more, please. So this is from a paper that we published. Again, I think this was from our wastewater microbiome paper. So what this is showing you is a fascinating thing that we found in one of our sink drains. On the bottom right is a cartoon of a sink drain. And in this sink drain, we found 2 organisms that were both KPC positive. Now, in this picture, so each of these kidney beans is supposed to be a bacterium, and the things that look like diamond rings are plasmids, and the diamonds, or the gems in the rings, are the KPC genes. And there are different KPC genes and different plasmids that are different colors. So we found in this sink drain, we found this Enterobacter cloacae, which had very strangely 3 different, so it's the beige one, yes, 3 different plasmids that had different KPC genes on them. 2 of them look the same, the pink little gems on the rings, and then the green one, which is a different KPC gene. And that's, if you just find that in isolation, you say, okay, well, there's a lot of plasmid exchange. Plasmids and bacteria are very promiscuous. We know that, especially in biofilm. And there's a lot of biofilm, a lot of nutrients in sink drains, so that happens. But what we knew was that a year earlier, in the upper right, we had happened to culture a patient in our surveillance cultures who'd been in that room, and we had actually also happened to culture. So we cultured the patient, and the patient's isolate had also gone into the sink. So the patient and the sink, at that time, a year earlier, had the isolate that you see there, which was a club pneumo, which had that KPC gene with the pink-looking plasmid and the green KPC gene on it. You see that same exact plasmid down below in the right that then went into the Enterobacter cloacae. So what happened there was the patient's club pneumo, maybe from like healthcare worker hand washing, got into the sink drain and stayed there. And through plasmid exchange, that patient's exact KPC plasmid went into an E. cloacae. But the even more interesting thing is that Enterobacter cloacae and the other two KPC plasmids that were in there, that we found in there, had actually come from a patient who'd been in the room even a year earlier. And it's just really coincidence that we happened to culture these patients. So a year earlier, on the left, we cultured a patient who had a KPC positive Enterobacter cloacae. And you can see from the colors and the looks of those KPC plasmids in that Enterobacter cloacae that's where that, and so it looks like it's in the sink, but it was also in the patient. That's where the other two KPC plasmids and genes in the Enterobacter cloacae on the bottom right in the sink came from, that we found a year later. So basically, this is just a really like live illustration of how these plasmids and bacteria exchange genetic material in the sink. And we just happened to catch it all in action as it was happening. So next slide. So Amy Mathers studied this further. We studied the exchange of genetic material. What she studied is how the organisms, I mentioned to you that organisms come from above, which is what I just told you. They came from the patients into the drains, and then they exchange genetic material there. She studied how they come from below, which is actually, even worse to me. We can control somewhat what we put into the sink, but we can't really control what comes from below. So in her sink lab at the University of Virginia, she studied how organisms get from the sink trap up to the sieve. Now, you don't want to think about that, but it really does happen. And this is where a lot of the patient safety hazard comes from. So in her study, which is really worth pulling up, the references there, it's really worth pulling up and looking at. In her sink lab, she put E. coli into the sink trap there, that's labeled, and put in nutrients for the E. coli, and then measured and watched as the E. coli marched an inch a day, all the way up to the strainer. And then you know what happens when you run the water. Next slide, please. When you run the water and there are bacteria on the strainer, this is what happens. So in this incredible picture, she put fluorescent labeled E. coli on the strainer of the sink, ran the water, and you can see that there's fluorescence everywhere on the faucet, on the handles, and all over the counter and the wall behind the sink. So this is how bacteria from the strainer get splattered all over, as far as a meter on either side of the sink, but certainly 18 inches at least. Which brings me to the next topic, I think you know where I'm going. These are pictures from my hospital. I have a bunch of pictures now, all in the same theme. So, you know, I think you've all seen the recent guidance from CDC, but it's not the first time you've thought about this. We want sinks to be deep enough to minimize splatter, and we want the water not to fall right on the sieve. But even when it doesn't, there's still some splatter. So even if we minimize splatter, there's still splatter. And if you have an incentive spirometer and clean towels and wound care supplies there, they will get contaminated with the bacteria that are definitely on the sieve of the sink. Next slide, please. This is another one, again, from my hospital. You've got IV bags and syringes and wound care supplies, a clean mask, the computer mouse. There's some medication sitting there on the computer platform, all within probably 18 inches or definitely within a meter of that sieve of the sink. Next slide. And now these pictures are from Danielle Rankin, who some of you probably know, or a lot of you. Again, incentive spirometer right there. Patient's going to put that in their mouth, and a lot of other supplies that are just couldn't be closer to the sink. And then in the picture of the faucet, that is oral chlorhexidine solution, which is ready to be contaminated from splashback on that sink drain and sieve. Next slide. And then finally, we've got pillows, syringes. It looks like open syringes for tube feeding, maybe, sitting in a cup there in the corner. Anyway, I think you get the message. We need to keep the counters clear because of the hazards of splashback of resistant bacteria that are almost always going to be on the sieves of sinks. Either they've come from above or they've come from below. And this is not an easy thing. This is not an easy edict to give to nurses or to any other healthcare workers, because there's just no space in rooms, and healthcare facility rooms have not been designed with this safety hazard in mind. And so this is a real design issue. There's no other place to put things, and it really puts nurses in a double bind, because this is unsafe, but they also don't have any other place to put things. And so we need solutions for this, whether they're splash guards or something else. Next slide, please. This is a study this is a couple of studies from Curtis Donsky at the Cleveland Veterans Hospital. He's a very inventive infectious disease physician. He's just constantly coming up with solutions to problems and studying problems. So he studied transmission from sink drains here. What he did was had researchers wearing gowns just like the one you see there, wash their hands or rinse their hands over known contaminated sink drains. So those were sink drains not with the worst organisms, but they had known gram-negative colonization of the sieve, and they weren't experimentally contaminated. They were cultured and found to be contaminated just in the hospital, like probably most of our drains are in most of our facilities. And then measured and then cultured the gowns, cultured hands, and cultured countertops. And so in the bar graph here, on the y-axis is the percent positive cultures, and let's just focus on the black bars. So the percent positive after running the water over hands, the countertop 11% of the time was positive for those gram-negative organisms. So 11% of the time you could find the gram-negatives from the sieve on the countertop. That's pretty bad. But from what you saw, what you've seen of Amy Mather's fluorescent picture, you're probably not surprised. Probably would have thought it might be more. The blue gown, was culture positive 9% of the time for the organism that was on the sieve, and then hands 6% of the time. Well, that's really great, isn't it? You're cleaning your hands. Now, they weren't using soap. They were actually using water in this case, but that you rinse the soap in the water, so you're washing your hands and you're contaminating your hands. So that's 6% of the time. Now, then what they did was they used that little gizmo in the bottom left, which is made by a laboratory by a little company that unfortunately stopped manufacturing them, but it was a very simple device. It doesn't block the drain. It sits on the drain by above the drain, just enough to allow water to drain down. The sieve is still there, and this sits over the sieve, and it's held in place by 3 suction cups. And it's just a light little plastic object that in the study they changed out every week or upon discharge, and whichever came first. And these are the clear bars, or the white bars on the graph. So with that in place, the countertop was culture positive 1% of the time, the gown 2%, and hands 0%. And the stars are there because these were not statistically significant differences because of low numbers. So, but in another study that they published, which is the first reference here actually, they found they also found less contamination of the sink when they did cultures of a sink when they used these little drain covers. So I'm not sure why they're actually not being studied further, why they're not being manufactured, but there may be actually problems with them that I don't know about, and I'm not trying to propose these particular items, but this is the type of solution that may need to be explored, or things like this that are simple, feasible and scalable to prevent splashback. But maybe things got stuck in these. I don't know what the feasibility problem was. On the next slide. So these are the CDC guidelines that just came out, and I think you may have all seen these. The URL is on the bottom right. And they have a terrific water safety in healthcare facilities website with outstanding advice and good references. So this, and the reason that this is such great advice is because we can't solve the deeper problem, which is disinfecting the drains. So the advice is, since we can't deal with the root of the problem, which is the drain, we can only handle the outside. We can clean and disinfect the sink basin, which we know is getting contaminated, the faucet handles, and the surrounding countertop at least daily. We know those are getting contaminated, so we can clean and disinfect those. We can mitigate the hazard that we know is being created by the drain that's in every patient room. We can try to avoid and come up with design solutions or retrofit rooms to avoid placing patient care items next to the sinks. And when we have designed new wards or refresh patient rooms, and we can select new sinks with faucets that are offset from the drains and deeper bowls that minimize splashing. They now recommend putting covers on toilets and hoppers. I wish I had videotaped my facilities manager when I told him about that requirement, because our toilets have no fixtures for putting on lids in my hospital. So that's going to be a real engineering job. But I'm going to go into that next and explain why that is recommended. And there's a very good reason. And then not discarding patient waste down sinks. So in Amy Mather's study, when the E. coli that was in the sink trap made its way up to the sink sieve 1 inch per day, it did that when it was fed nutrients. And nutrients are things like Ensure, Boost, Coca-Cola, D5, D50, glucose, liquids with glucose. And so if we put nutritional supplements and beverages down sink drains, we are feeding those bacteria and giving them just what they need to rise to the sieves. When she did not put those nutritional supplements down the drains, the E. coli actually did not levitate like that. They stayed pretty much stagnant in the sink traps, or if they did rise toward the sieves, they did so much more slowly. And then not putting patient waste down sinks. That may be fairly self-evident, but it probably happens. May be self-evident to you and me, but may not be to everyone. So it is listed in the website. Next slide, please. So let's talk toilets. This is where it gets real. So, you know, there's a reason we don't put our purses down next to public toilets, or really any toilets. We hang them on a hook as far from the toilet as possible. And it's because we intuitively know that, you know, there's an aerosol that follows a flush. We all know that. We've all also seen studies about bioaerosols from toilet flushing. This is a study that came out from the CDC epicenters in 2018, and I really admire and pity the people that did this study, because they had to go around a hospital and get paged every time someone had a bowel movement, and then measure the both particle counts and bioaerosol levels when they flushed the toilet. So, but that's, you know, that's science and progress, and now we're talking about it. So, you know, they contributed to the medical literature. And so in this study, they compared the bioaerosol concentrations as far away as 2 meters, so 6 feet from the toilet, under conditions of no flushing, so nothing happening, flushing an empty toilet, and then flushing a toilet that had stool. They also measured particle concentrations. So particle concentrations, I'll talk about in a second, but bioaerosol means like the live bacteria that you can culture. So that uses culture plates. So, and that's probably more what we care about. So when there's no waste in the toilet and you're not flushing the toilet, there's a certain bioaerosol concentration in the air in a bathroom, and that you can sort of imagine that it's a bathroom. When there's no waste and you flush the toilet, that bioaerosol concentration, you can see it goes up from 210 to 240. Not as much as you would think, though, right? You think it would go up a lot more than that, which means that's a rather high baseline. And then when there is stool in the toilet and you flush the toilet, it goes up some more. Well, that's intuitive. That's exactly what you think would happen. And that's 6 feet away. And in fact, the researchers captured that 6 feet away, and they didn't go further away than that. So, you know, you think about what is 6 feet away from toilets in patient rooms. And in an ICU, there is often a ventilator much closer than 6 feet, or other patient care equipment closer than 6 feet or 2 meters to a toilet. There's often a yank hour sitting on a ventilator closer than 2 meters from a toilet, for example. And then the particle concentrations, interestingly, increase more after empty toilets were flushed than toilets with waste. And that may have to do with sort of dampening of the, well, it's just gross, but dampening of the material in the toilet, and having it sort of weighed down by gravity. But we don't have to go there. the point here is that the even 6 feet away, they were able to measure this live bacteria in the aerosol. And I wonder what they would have found if they even went further away, but they didn't in this study. So next slide. To that point, Amy Mathers, again at the University of Virginia, had a problem in their ICUs there. They had a number of ICUs. In 5 ICUs over an 18-month period, they had 56 acquisitions of KPC organisms. And 20 of those were in clinical cultures, 36 were colonization. And their genome sequencing suggested that there was, rather than patient-to-patient transmission, it suggested a nosocomial reservoir, particularly environmental reservoir. And they were culturing these hoppers, which are like toilets, but they have a higher velocity flush. And these are in ICUs. And they look just like, this is a picture from their paper. This is what the hoppers look like. And they had this sort of separator on the left, and then they had patient care equipment, that was definitely within 2 meters of the hopper. And the nursing staff would pour waste material into the hopper and flush, and it was a very high velocity flush. The study I just showed you was regular toilets, not hoppers, with those bioaerosol concentrations, so it's probably a lot higher with the hoppers. So on next slide, go back, please, I'm sorry. So what she did was, having cultured some of the same organisms from the hoppers and the patients, she did a before and after study, a quasi-experimental study in which she had her facilities folks manufacture lids for these hoppers, which look like the picture on the right. And so they added lids, and then she followed the rates of infection and colonization in those ICUs. Now, next slide, please. And what she found in comparing the 18 months before the lids went on to the 18 months after the lids went on was a significant drop in the rate of KPC acquisition. So they went from 56 to 30. Clinical cultures went from 20 to 9. And the rate of clinical cultures per 1,000 admissions went from 9 to below 3, which if you click again, you can see is a drop of 70%, which was highly statistically significant, which is phenomenal. And that's from putting lids on the toilets. I mean, when it's just you can't really expect to get stronger evidence than that of an infection control intervention. So, the unpublished data that I've heard from this site since then is that they've actually had further reductions following their rates out beyond those 18 months. That their rates have continued. This wasn't just a fluke. Their rates have continued to drop, which is wonderful. So this actually is the basis for that recommendation on the CDC website. And there may be other sites that have done this, but this was a major source of the data. So on the next slide, please, I will conclude by just reviewing that drains, toilets, and other wastewater reservoirs are very clearly documented sites of direct or indirect transmission of multi-drug resistant organisms to patients. Given that, the best we can do right now is to optimize hygiene practices, environmental cleaning, and infection control measures. But we need innovative, yet feasible, cost-effective, and scalable solutions for this patient safety problem. So one of you out there, please go out and invent something. There actually are companies and academics working on different solutions, and I didn't have time to go into it, but there are a number of papers in the last year and a half describing different gizmos that use vibratory mechanisms, heat, electrical currents, all types of things that go around sink drains to try to disrupt the biofilm. And they're all experimental studies. None of them have patient outcome data or any kind of meaningful clinical outcomes, to say whether they're going to be promising. But people are trying to work on this. But what we need is something that will not cost, $10,000 per sink drain or even $5,000 per sink drain. I think the cost-effective part of this is going to be very important. But I don't think we really even know what the human cost of this problem is, because we're really just beginning to appreciate it. So thank you very much. And I don't know, is it possible to take questions?
Hi, yes, absolutely. Thank you so much, Dr. Palmore. Now, I'm pretty sure probably more than half of us don't want to go to any public restrooms anymore, I mean, thank you. That was absolutely
You're
amazing, and it
welcome.
was and it was great to hear a lot of the background and studies that were conducted to provide those CDC recommendations. So if you guys have any questions, please go ahead and type them in. I wanted just to quickly address, I know that you said that this has been studied for some time, that for about 25 years we've known that this could be waterborne pathogens, sink drains, could potentially be a source of contamination and infection for our patients, but not till recently, for the past 5 to 10 years, has that changed. I want to know from your perspective how long you think it'll be until sink hygiene really becomes a Category 1A recommendation through CDC, because that's really one of our barriers to adopting a lot of recommendations is, we're really looking for substantial data and being able to say, yes, absolutely, this is something that we need to adopt into our healthcare facilities. So I just wanted to get your input on that.
Oh, that's a great question. I don't know how long it'll be. I think that there probably will need to be more studies and more data. I think that it's been suspected that sinks were a problem for a lot of years, but it's really only been in the last 5 years that there was convincing evidence. And things like lids on toilets, there's maybe one published study now that shows that, and I don't know that will become a requirement on the basis of one study. So I think there will need to be more. There will need to be a bit more evidence about some of this, and then also better solutions, because I don't think that there can be mandates without clear solutions, and we don't have a lot of solutions. But I think for some of the basic elements like wiping down sinks and the environmental cleaning aspects of this may find their way into some of the existing environmental cleaning standards. That's what I would expect. But I don't know. I don't have any special insight into that.
I was hoping you would have the scoop for us, but that's okay.
No, I don't have any inside track on that. Sorry.
Okay, so we have a comment from Crystal Bayer. She says, I was completely fascinated by the fact that we may be feeding pathogens by dumping nutritional drinks down the drain. All I can think of was the movie Blob. Now, I think that that's a fantastic comment, because I've actually presented to leadership at my hospital about this, and I spoke to them about, these are the recommendations that have come out. These are things that we need to look at. And when I got to the part about we really need to not be putting D5 down the drain or be putting, other types of solutions containing glucose or drinks, I immediately got a little bit of pushback of, well, where are we supposed to dump it then? Where is this supposed to go? Because we need to get rid of it somehow. And I just wanted to know how you have dealt with that at your facility and what type of recommendations or advice you would give to other infection preventionists who may be trying to really implement this at their facilities.
That's awesome. The blob. You've hit on exactly the problem, which is that there aren't really great alternatives. So people can, like, in theory, you can throw things out with a thickener, but that's time consuming and also messy and kind of impractical. Um, but so that's the type of thing that uh, needs solutions, and I think with a little bit of passage of time, um, people will um, people will come up with solutions for that. I mean, right now, what we've done is we end up um, just tossing out like the rest of the bag kind of full in the trash, which is not a good solution. Or if there's a choice, it gets dumped out in the toilet rather than the sink, which is also, again, it's on the website. It says don't put it in the sink or the toilet. But if we have to choose one, we put it in the toilet because that's that's already where waste is going and it's already super contaminated. So, we have to make hard choices. We have to sort of pick our poison, and it's a catch-22. So yeah, I think for a lot of these recommendations, they're really good recommendations and they're absolutely right. But it's hard to know what else to do. Like, it's hard to know where else do you put a pile of pillows when you don't have any shelves in a patient room. Like, so
Yeah.
we have a ways to go in coming up with solutions, and that's why I think it'll be a while before there are mandates.
Perfect. We have a question here from Frances Vaughn. She says, do you think splash guards are effective?
I don't think they're that effective. I think that um, they're because I think what happens is things just get piled up on the side of them, and sometimes higher than the splash guards. But I think in some cases they're probably they may be better than nothing. I think it's a facility by facility choice. So I'm not recommending splash guards. I think the ideal is to um, is to find a design solution to keep that counter space free. I think that's actually the best solution. Splash guards fall apart, and also sometimes the stuff that sticks them in place gets uh, gets dirty, and um, splash guards also don't get cleaned properly. There are a lot of problems with splash guards depending on the facility. So I think it's problematic to recommend splash guards across the board for everyone.
Perfect.
And I,
We
I
have
have seen, I didn't have a picture, but I have seen one facility. I didn't feel comfortable taking a picture where there were splash guards, and then on the side of the splash guard was a pile of patient care materials that went up like at least 6 inches higher than the splash guards, which sort of
Oh,
defeats the purpose.
yeah. We have another question from Hassan Abdallah. Thank you for your great lecture. Did you find any resources talking about sinks as a source of Candida outbreaks, or more specifically, Candida auris?
Sinks, not yet. I have not seen any data or heard any data about Candida auris, or um, I think sinks are rarely a source of Candida outbreaks, but there are a few older reports of Candida from sinks. But generally, Candida get outcompeted in sink drains by bacteria.
Perfect. So we're a little bit over our time, so we are going to go ahead and conclude. But before we go, Dr. Palmer, if there was just one thing that you would ask for us infection preventionists that maybe we could start focusing on in our facilities, I know that there are many different, recommendations that have been out, but if you think there's one that we should really try to focus on the most, which one would it be?
You mean of all of what we talked about today?
Yes.
I think it would be clean. I think it would be the cleaning. I think that's the thing that of all the things that are recommended, that's the one that we all can just do right now. We can't all right now clear out 3 feet of space around our sinks. We can't all stick lids on our toilets. We can't all, necessarily figure out where to dump glucose solutions, but we can clean. We can all start cleaning in around our sink bowls, and so that's I think the thing, the low hanging fruit. And
Perfect.
thank you
Well,
very much for inviting me to do this. I enjoyed it, and thank you for your great questions.
oh no, we appreciate you taking the time out to, educate us and help us become better infection preventionists. So thank you so much for your time. All right, guys, this is going to conclude our November group lecture meeting. So thank you all for coming. And just so you know, Jillian is coming back in December. She's going to be doing carbs and carbapenems, holiday special. So we'll be talking about carbapenems in December. Okay. Thank you so much, Dr. Palmer, and y'all have a great weekend.
Thank you. Bye bye.
Bye.